So Daniel, how are you? Is it? Yes, yes, we just he's translating my book. Yes, very good idea. I had an email from one maid who said she couldn't come. Even tonight, that was for dinner. And next week we will be coming to the NRPB symposium. So we'll be there. Do I get more money? Yes, yes, yes. I was joking. Every time I sign. I like to stay.
各位女士、各位先生，我们要开始今天的一个 special lecture。这个同仁可能他们如果开始之后进出可以从后门啊。So it gives me a pleasure to introduce our special guest, our speaker today, Professor Bertrand. He has been a long-term friend to Taiwan in supporting from the era of conducting the national research program on genetic medicine, and then now the NRPB. He is a very famous scientist in the field, and also he has deep humanity and social science concern. So he's also acting as our ELSI director of the entire Human Genome program, and then later the NRPB program. And I personally has been given this privilege to discuss and also co-publish some paper with him. We wrote one article on autism, the French story, and also the ethical, legal, and social impact concerns long ago. But still, we are receiving advices and also very good guidance from him. And Professor Jordan, he is currently the NRRS Erasmus Research Director, though he retired. And he also has been in charge of the Marcelli mice genome pool research. And currently, he is the counselor of our NRPB program. So today, his talk will be ELSI issues in clinical new generation sequencing. Let's welcome Professor Jordan. Before I give this microphone to him, I also like to introduce two special guests. Okay, one is Professor Jim Dwyer. He is a visiting professor to the Department of Social Medicine. He has been here for 1 year as a visiting professor. He teaches medical ethics and cooperates with us in many research programs. Okay, this is Jim. And the other one, we might be surprised, is Mr. I know your name in Chinese, Lin. Okay, he's a journalist. Okay, Lin Changhong. So in Japanese is Masahiro Hayashi. So he's a journalist, yes, and he is also a fan to Professor Jordan. He has translated his 2 books from French to Japanese. Okay, and then he flew from Japan this morning, especially to join this meeting. So let's give him a warm welcome.
Thank you very much, Daniel, for giving me this opportunity, and also giving me the opportunity to meet my Japanese translator, which is a big pleasure. Yes, so every year I come to Taiwan, and every year I have a discussion with the ELSI people, with you, on a topic of current interest. So, given the development of new generation sequencing, I thought this would be an interesting topic for you. So, of course, I am not an ELSI person. I have some interests in the field, but I'm not a specialist. So, really, my talk will be probably two-thirds telling you what's going on with new generation sequencing, and one-third discussing the issues it raises in terms of ethics. So it's not really a 100% ethics talk. But I think, given the way this is moving so fast, I think it's really useful to know where we stand today. So let's see if I can operate this thing. Wrong button. Wrong button again. I want to move forward. These things can be temperamental. Let's see if this works. No.
Here.
It's not going to work. I think it's too far. Okay, I will stand on this side. Let's see. Yes. Now it works. Okay, so I'm starting, as I said, with an update on new generation sequencing, NGS for short. Oops. Okay. Sorry. Okay. This is a map you can find on the internet. It's a site which gives you the number of new generation sequencing machines that have been installed worldwide. It's quite impressive because this was introduced maybe 4 or 5 years ago, and these machines are about $500,000 each. And as you can see, it has really made a lot of them have been sold now, being used of course in the United States, in Europe, and also in the East, in Asia. And if we zoom in a little, you see that you have quite a few actually in Taiwan. The figures on this site are not exact. I mean, it's underestimated because it's based on what people put on the site, and not everybody indicates. So there are more than that. But you can see, for example, that compared with Japan, Taiwan is doing quite well in terms of being involved in this new field. Because as we'll see, the increase in performance is so big that it's really opening up many new fields. And I think I have a close-up of the Taipei region, and you see a big cluster here, which corresponds, of course, to Academia Sinica and its satellites. Yeah. Okay. Okay. So that's
It's hard.
It's not working. It have to be close. It only works if I'm close to the gadget.
Maybe do this one.
Okay, thank you. Yeah, I mean, I gave talks in Tainan yesterday. It was simple, so it's not specific. So, as I said, this technology has really caught on, as you saw from the number of machines that are being used worldwide, and it has become essential in research, in academic research. It's actually replacing microarrays, which has been my pet subject for a few years. Not in all applications, but in many applications. And it's really opening up something which one might call sequence-based biology, a way of looking at biological systems in which you know that you can get any sequence, any amount of sequence of any organism very quickly and base a lot of conclusions on that. And it's also having an impact in clinical research, in working out monogenic diseases for which the gene was not known, in trying to understand what's going on in cancer, actually finding that it's even more complicated than we thought. but what I want to tell you about today is whether it's really now already being applied in clinical research, not in, in clinical practice as a diagnostic in hospitals to treat patients. So next slide, please. Just a few words about the hardware, the machines. Next one. Even close by. So at this time, there's basically two classes of new generation sequencing machines. There are large systems, and as we see, there are smaller systems. So the large systems, there's essentially 3 or 4 major manufacturers who are competing very hard, which is part of the reason why the field is moving so quickly. And so these are machines that cost around $500,000 US, so pretty big pieces of equipment. But they are able, in just one run, which lasts a few days, they are able to read 500 gigabases of DNA, 500 billion bases of DNA, which translates, if you take into account the fact that you need to read several times to have reasonable accuracy, which means in just one run, one of these machines can read several human genomes. I mean, which is a tremendous change. I mean, if you remember that the Genome project lasted 10 years, costed several billion US, involved hundreds of labs all over the world just to read the human sequence once. Now with these machines, you can read 3 or 4 or 5 sequences in a week. And the cost is about $10,000 US for one run of these machines, so which means a few $1,000s per genome, not a few $1 billion per genome, as only 10 years ago. Okay. So it's really such a change by several orders of magnitude, so really changes the picture. So these are the large systems. And now, not a new generation, but a recent development is that now there are small machines which you can put on the bench top. Next slide, please. There's actually 3 currently on the market. They look more like this. Fairly compact machines, which you can buy for 50 or 100,000 US dollars. So it's no longer a very big investment. They're also much faster. They do their run in 1 hour, 3 hours, 5 hours, depending on the machine. So you can get quick results. Of course, you get less sequence data, and it works out to be more expensive on the per megabase data, per megabase reckoning. But it's still much cheaper than the conventional sequencing. And it may be that these machines will actually open up the clinical field. I mean, that's can be debated, but anyway, this is a fairly recent development. And actually, development is still going on, as shown in the next slide. Now there is a next generation coming up, which is based on nanopores, so-called nanopores, where basically you manage to get the DNA to go through a membrane, and as the DNA goes through the membrane, you manage to read the bases one by one, and in this way you can get a lot more sequence, a lot cheaper, a lot faster. Several companies are developing this. One of them announces a gadget like this, which will supposed to cost $1,000, and which you plug into your computer and which reads a few megabases of sequence. It's a disposable device. So this may be true. It may come in 1 or 2 years from now. So it's still. The technology is definitely not stabilized. There is still a lot of progress going on, but people are using it a lot. Okay, next slide. So what about the clinic? Next slide. Sorry, I have many slides. So that's the question. This is a drawing I quite like, a cartoon I quite like. So it shows a patient who's not really feeling good, and his doctors who are reading his DNA sequence. So they have all his DNA sequence, all 3 or 6 billion nucleotides of it, and they cannot make much out of it. It's very interesting, but what use is it really for treating the patient? So that is the question. Are we already able to use this kind of information in the clinic? Next slide. So one can actually, there are indeed a number of current clinical applications which one can put in 3 categories, if one likes. This one, full exome sequencing. I'll explain what exome is, mostly for mysterious cases where you're not able to find out what is wrong with a kid, although you're pretty sure it's a genetic disease. There is, of course, the cancer field, and there are a number of approaches applied in either in pediatrics or also for screening, preconception, or maybe pre-implantation. So I'll briefly describe these 3 applications and give 1 or 2 examples. Next slide. So this is an approach that has already been used in a few hospitals in cases where you have a child with a disease which may be mental retardation, dysmorphism, whatever, muscular dystrophy, but you're not able to find out which is the defective gene. Maybe you have tried the most obvious possibilities like Duchenne muscular dystrophy and a few others, and you have found no mutations. You don't know what's going on. And one approach that has been successfully used in a few cases is then to do full exome sequence, which means sequencing all the genes of this in the DNA of this individual. Not all the DNA, as you may remember, in human DNA, the genes themselves actually only occupy 1 or 2% of the DNA. There's a lot of junk DNA, so-called junk DNA, pieces of DNA between genes and within genes that play no obvious role. Most of the useful DNA is in the so-called exons, the expressed parts of genes. And there are now methods that work quite well and that are quite inexpensive to take a sample of DNA from a blood sample from a patient and extract from this DNA only the gene coding part, only the so-called exome, the sum total of all the exons. And of course, the advantage is that in that case, that means you have to sequence maybe 100 million bases instead of 6 billion bases. You reduce the amount of work by a factor of 100, just about. And so what can be done is to do this trick on DNA from the patient, then sequence this DNA very accurately, which means you read this DNA maybe 100 times so that you are almost sure that there is no remaining mistake, because of course the problem with all these techniques is that you have a certain percentage of mistakes. But if you go over DNA many times, you minimize the number of mistakes. And then, of course, you have to do bioinformatics. Now you have the sequence of all 25,000 genes from this individual, and you use various methods to see if there's anything that might explain the condition. And I think the next slide shows one example. So yes, this was a small child with very severe autoimmunity problems. Well, they were thought to be autoimmune problems. And he had fistulae, stomatal fistulae opening to the outside, making operations necessary. He had already been operated many, many times, and then the problem was continuing. So because this looked like an immune problem, so a number of genes to be involved in immune or autoimmune diseases had been tested, but no mutation had been found. And so finally, they decided to do whole exome sequencing to look at all the genes in one at a time. And handling this sequence data, they found indeed a mutation in a gene which is called XIAB, which is an inhibitor of apoptosis. So apoptosis is a mechanism by which cells commit suicide, if you want. And so an inhibitor of apoptosis is a molecule that prevents cells from committing suicide. And so you can imagine if this inhibitor is missing or is faulty, that you have the kind of symptom that this child presented. And since this gene was known to be expressed in blood cells, so this indicated that bone marrow transplantation, providing a different type of blood cell progenitor could help, and indeed this was performed, and the boy is doing well. So this is one case where the testing likely hypothesis one after the other did not provide the answer, and being able to do all of the 25,000 genes at one go gave you the answer. And of course this was completely impossible 5 years ago. Next slide is now cancer. So what is now, quite currently being done is to not to look at the whole cancer genome, not to look at even at the whole cancer exome, but to focus on a few hundred genes that are already known to be often involved in cancer, in various types of cancer. In fact, we're finding out that there's not much specificity in each type of cancer. So you purify or amplify the coding part from 100, 200, or 300 genes that are already known to be often involved in various cancers. Again, you sequence this to high accuracy, and you look for known mutations or for unknown mutations in known genes. And there, what you hope to find is mutations in genes that code for proteins for which there is a drug already available, either already marketed or maybe undergoing clinical tests. So the reason why people take this this focused approach is the incredible complexity of cancer. Maybe next slide. Yeah, why not just sequence the whole genome of the tumor and derive conclusions from that? And the problem is that when you do that, next slide, when you do that, you get something like this. This is a paper published maybe 2 or 3 years ago, and it shows the results of sequencing a lung cancer, small cell lung cancer, and it's actually the comparison of the sequence of DNA in the tumor compared to the DNA from a normal cell of the same patient. So it's really, well, it should be cancer-specific differences. And the problem is that there is an enormous amount of differences. You have lots of insertions, you have lots of deletions, you have lots of substitutions, you have copy number changes, small regions that are amplified or that have disappeared. You have rearrangements inside chromosomes. With the green lines, you have rearrangements between different chromosomes. You have a whole zoo of alterations, and you don't know what is important and what is not so important in this. Next slide, please. So actually, in this tumor, as stated in this paper, there were 22,000 substitutions. As the authors wrote, one mutation for every 15 cigarettes smoked, because this patient was a heavy smoker. I mean, it's a pretty good deterrent. Every time you smoke a pack, bang, you have one mutation. And when you have enough mutations, you get sick. And so you don't know which of these mutations are drivers, are really driving the cancer, and which of them are just happen because the cell is just all messed up. So it's better to have a focused approach. Next slide. So for example, it is known in many types of cancer that one of the drivers of the cancer is often a kinase, an enzyme which adds phosphate, which is deregulated, and there are indeed a number of drugs that are kinase inhibitors that inhibit the specific kinase that is overexpressed in a given cancer. And so if by doing this sequencing you find a mutation in a gene coding for a protein for which there is already an inhibitor, you know that this is a drug that is likely to be effective on this patient at this stage of development. So next slide, please. So as I said, this is already in clinical practice, and some companies are offering this type of test. This company called Foundation Medicine using next generation sequencing because you need a lot of DNA sequence. You couldn't do it by conventional methods. So their test looks at 200 cancer-related genes. They can actually prepare the DNA corresponding to these genes from a sample, even from a sample that's already been put into paraffin blocks, which is what is usually done in hospitals. And they sequence it to very high accuracy. They read each position 500 times to be sure of the results. And from the experience they have so far, they found several what they call actionable mutation. Actionable mutation means a mutation that tells you that there is a possible therapy, tells you that there is an inhibitor for what is caused by these particular gene alterations. So they do get useful results in most cases. They do the test in something like 2 or 3 weeks, which is not too long. And as you see, the cost is fairly high. It's very high for a genetic test. But on the other hand, when you know that a typical cancer treatment may cost $50,000 or $100,000, and that by doing this, you're going to use the right treatment for the right patient at the right time and avoid unnecessary treatment and unnecessary side effects, it's certainly quite worthwhile. So this is an approach that is being quite widely used. Well, maybe in the future, people will do whole exome when it becomes cheap enough to make sense, but at this time this is widely done. Next slide, please. And so the third type of application, well, of course, these applications are not completely different, but they have some specificities. Again, this focused on known mutations, so-called Mendelian mutations. As you know, there are at least 1,000 genetic diseases that are caused by a defect in a single gene. I mean, a different one for each disease, but and in which we know which is the gene. Each of these diseases is quite rare, but collectively they account for, I think, something like 2% of births. So usually when you have a child, with such a disease and you don't know, well, you only have you don't know which gene is involved, you make a guess, you have a genetic test, and you find whether it's confirmed or not. And there are, at this time, there are many such tests on the market. The next slide, I think, shows that. This is the growth in the number of DNA tests available in the United States, so it's now more than 2,000. So each of these tests looks at one specific gene, maybe at several mutations in one specific gene for a given disease. A typical one is cystic fibrosis. So cystic fibrosis, we've known the gene for almost 20 years.